Welcome back to the NPTEL lecture series on animal physiology. So, we are in the section of kidney and the regulation of fluid, or in other words, in the excretory system. So, we have already done the basic structure of the smallest functional unit of kidney, the nephron, how the individual nephrons are involved in the whole filtration process. So, we have done with the structure, but a little bit of our structure is left, because when I drew the structure, I showed you distal tubule, proximal tubule, Bowman capsules and all those things. So, what we will do initially in our first slide, I will explain all the different cellular structures which are present at different zone, at proximal border, at distal border and all other places. And then we will talk about the simple driving forces which are regulating this whole process, and then we will talk about the counter current multiplier, and we will close in with the renin-angiotensin system which is regulating the fluid. So, even before I start with the cellular structure, let us have a simple understanding exactly what is happening. The way it works is fairly straightforward if you look at logically. So, the blood which is coming, so blood is not in physical contact, not in connection with the nephron. So, basically here is the blood vessel which is entering, and here you have the nephron. If you go back to the structure, you will look at it. So, there is a gap between the two, fine. So, say for example, this blood vessel is carrying something, say molecule X, okay, whose concentration is higher, and whereas the concentration of the molecule X in this vessel, in the nephron, is lower. So, automatically from the higher concentration, we will move on to the zone of the lower concentration. So, one of the forces which promotes this whole process is osmotic pressure. And the second force, you have to realize there is another force which is governing here, that is the hydrostatic pressure, the amount of water which is present on these blood vessels and the amount of water which is present out on the other side into the nephron, because it is the water which is carrying the ions. It is not that single-handedly just the ions will move out. So, whenever the ions are moving, along with them, they are pulling a stream of water with them. Okay. So, hydrostatic pressure and osmotic pressure, we will talk about it, and we will talk about one of the very interesting mechanism called counter current multiplier, which is fairly common in biological systems, but we will just kind of go through it, and I will expect you guys to consult other advanced textbook to know more about it. Okay. So, let us continue, and let us talk about the different cellular structures which are present in the proximal and distal tubule of the nephron. Okay. So, coming back to the slides. So, basically in the proximal tubule, cells are like this. Proximal tubule cell. So, these individual cells have a lot of brush borders like this. Okay. These are the brush border, your toothbrush, if you look at it, it is almost like this. So, this is the brush border which is increasing its surface area of contact, and here it is. Okay. This is the basal part of it. Okay. And you have of structures like this all over the place. Okay. Okay. So this is how the proximal tubule cells look like, which forms the proximal tubule of the kidney, and these structures are nothing but mitochondria. A lot of mitochondria, there is a lot of energy dependent processes are happening, and underneath you have the basolateral labyrinth actually. Basolateral labyrinth, and this is the brush borders. Okay. So this is all about the structure of the proximal tubule. From here we will move on to the structure of the distal tubule cells. Depending on their functions, their anatomy changes. Distal tubule cells. So the distal tubule cells are something like this. The basic structure remains the same. You have the mitochondria likewise, but the stark difference comes somewhere else. So they hardly have any brush borders. Okay, so there are no brush borders, and you have of course the mitochondria sitting here like this all along the place, and you have the basal membrane. This is the basal membrane. These are the MT stands for mitochondria, and these cells are without the brush border. And then there is the third kind of cells which are present in the loop of Henle. Loop of Henle cells. So these loop of Henle cells are something like this, which is totally different from the distal and the proximal cells. Okay. So after looking at these cells, you will be able to appreciate that how a different part of the whole nephron, the cellular architecture changes. You cannot, if there is no uniform cellular architecture, and cellular architecture changes based on the requirement of that particular part where what is happening. Okay. So now I will just draw another cellular structure to show the epithelial cell lining between the glomerular into the Bowman capsule and the blood vessel, how they are doing. So coming back to the slides, let's draw the glomerular membrane. Glomerular membrane. So essentially this glomerular membrane is something like this. Okay. Okay. It's kind of a perforated structure out here, what I am trying to draw. Okay, and back to the frame. Okay. So on this side, so you have the blood on this side and the glomerular filtrate on the other side. Glomerular filtrate. Okay. So what is happening essentially is these are the RBCs which are there. You have the plasma proteins on them. So RBCs, just putting PP as a plasma protein. So this is the endothelium lining of the blood vessel. Endothelium lining of the blood vessel, and these are the fenestrated capillaries, if you remember, fenestrae or the windows, and on this side, and these are the slit pores are kind of like this. Okay? Not fully continuous. There are certain barricades out there. On this side, you have the epithelial lining of the glomerular filtrate, glomerular membrane, and these are called the slit pores. And essentially, what is happening is these fluids are moving like this through this, maybe in an energy dependent manner, maybe in an energy independent manner, likewise. So this is how the movement of the fluids are taking place from the blood vessel to the glomerular membrane. So there is essentially what you do, you put a physical mesh around, and through the mesh things are moving. Though they are in physical contact, but they too are different entities. It's like one hand and this hand. They are two separate entities, but in this junction what I was trying to draw in the previous slide was, in this junction all the movement of the fluid along with the solutes is taking place. So, and this is a very common thing all across the biology, where you see two individual entities. So, there is nothing like a gap junction or something. It is, there are small pores, and those pores are regulatory pores. They are not just physical pores that anything can pass through. This diffusion, what I just now highlighted, could be energy dependent diffusion, energy independent diffusion, protein carrier mediated diffusion. We will talk about all those things, how these transport is taking place, but this is a simple transport phenomena which either may need energy or may be independent of energy requirements. Okay. Now, from here I will move on to the next, which is the Bowman capsule. Back to the slide. So talking about the Bowman's capsule. So, Bowman's capsule is something like this. Bowman's capsule. Okay. So, the Bowman capsule is, if this side is showing this is the Bowman capsule. Okay. This is the membrane, and okay. On this side you have, the blood vessels. So, the blood vessels are coming like this. They are entering like this, and there is a lot of networking of the blood vessel. Then they are almost, like this. It comes out like this. So, back. So, this is your the arterial blood entering, or the afferent arteriole. Afferent arteriole. This is the arterial blood. Okay, and you have the glomerulus membrane here, and what is essentially coming out is the, this is the glomerular filtrate, and this is, you have the Bowman capsule. So here you have the Bowman capsule. These are efferent arteriole. Okay, and this is your glomerulus, and this is the glomerular filtrate. So, after this basic understanding of the structure, I will move on to the next part, which is the forces which are driving this whole filtration. So, as I was mentioning just in the beginning of the lecture, there are two major forces, hydrostatic forces and osmotic forces. So, let us look at it and let us enumerate all of them, and let us see the relation between them. Okay. So, coming back, what are the factors determining the glomerular filtration? Right. Okay. So, the two major factors which are, so this is also called GFR, This is regulated by two forces. One is the hydrostatic force and hydrostatic force is essentially between the capillaries and here you have the kidneys, nephrons, nephron vessel, individual nephron. So the hydrostatic force, something like this, if represented by an arrow. Then you have the next force, which is the osmotic pressure. You can also call it hydrostatic pressure or force, or osmotic force or osmotic pressure. Okay. So the relation stands like this. GFR is equal to, K is the constant, okay, C yes, PI. I'll just explain what is that. Pi C minus pi I, where C stands for capillaries, P stands for hydrostatic pressure, this P also stands for hydrostatic, I stands for Bowman capsule, this I also stand for that, and this C stands for capillaries, and this pi stands for osmotic pressure. So the difference between hydrostatic pressure and the osmotic pressure is considered as the glomerular filtration rate. So this is how at different zone the filtration process is being executed while the fluid is moving through it. And whatsoever fluid initially moves at the Bowman capsule is being absorbed, reabsorbed and secreted, and likewise it goes through a whole channel. And the whole idea is to retain as much water as possible and not to lose the electrolytes. So electrolytes have to be retained, and in that process getting rid of the urea and uric acid and all other unwanted molecules which could destabilizes our homeostasis of the body. Okay, so from here we will move on to, so let's coming back to the slides. Okay, so let's get back to the slides, and so after this we will just move on to the next part of it, which is your, just give me 1 second. We will talk about how the sodium is getting absorbed at different zone. Next slide. So how about this? So we take an individual case of an individual nephron, and we divided it into three parts: the cortical region, the cortex, outer medulla, and the inner medulla. So this is the cortex, this is outer medulla, O stand for outer and I stand for inner medulla. I will draw the nephron now. So the nephron is like, out here it starts and it is moving through. So this is the distal tubule, and it is all the way moving down, 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 and then again it's becomes thicker, and then coalesce, and here the urine is getting formed. That's it. So if you look at it, so this is the zone where it is much more thicker. This is the thicker segment, which is then it becomes thinner. This continues to be thinner, and then again it becomes thicker here. The Okay. So, likewise, this is how it goes. So, now what is essentially happening is this. So, this is how, let me just make it a little much more clear. Okay. And now use so, here the blood vessels are entering here. Okay. Now, coming back. Okay. So, basically what we will be talking here is about the sodium resorption in the nephron. So, this is one of the major molecule which you have to ensure that it is regulated right in the nephron. Okay. So, the first thing which is happening, so sodium is entering through this. Here the sodium is coming in. This is all in the blood. So, from the blood, the sodium molecule. So, if I represent the sodium molecule, so now let me represent the sodium molecule by, say, I use, for example, I use the yellow color. Okay, these are the sodium molecules. Fine. So, these sodium molecules then first move here. These yellow colors are the sodium molecules. So, they are traveling down. Fine. Concentrated out here. So, at this point, somewhere out here in the cortex, part of the sodium is being using a energy driven process, part of the sodium is being reabsorbed. So, here it is getting reabsorbed. Fine. So, then again sodium is moving through out here, and if you look at the osmolarity value of the sodium, you will see at this zone, this is 290. Okay. So, the value went down further. So, this is 100% which has moved out here. Okay, then it's 290, and then out here sodium is moving all the way. Okay, all the way through the loop of Henle and out here again at this zone. Second level of sodium absorption is taking place, and this is again an energy driven process. So, this is an energy driven process, again sodium is getting out. Okay. Then again sodium is moving all along this, all along this tubule, and out here this part of the circuit is very important. So, I will highlight it with another color just to tell you, like this is the zone which is under the control of the aldosterone. This is the zone. This is purely under the aldosterone control. Okay. And the third level of sodium absorption takes place at this point out here. Out here, the third level of energy dependent sodium absorption is taking place. The last level where sodium is being again retained by the system, and osmolarity values of sodium keeps on changing from 290, it becomes around 600 here, the maximum is here 1200, and then out here it becomes 220, and likewise it keeps on changing. And at this zone is called, this is the zone where, one second, okay, this is the zone, the shading zone, what I am doing with the shade. Now, this is called, this part is called isotonic resorption. Isotonic resorption. So, this is how the sodium, so if you look at it, the most simplest of all, the sodium is kind of reabsorbed in a very interesting manner as it is moving through, and all the time it is the two driving forces which are playing critical role, the hydrostatic force or hydrostatic pressure and the osmotic pressure. These are the two key players which are regulating this whole process very tightly. Okay. From here we will move on to something called counter current multiplier. What is, because if you realize these tubes are in close proximity. So, if I am drawing this thing with a lot of space, but in real life they are fairly close, this tubes, loop of Henle moving. So, what is counter current multiplier? So, counter current multiplier is, say for example, you have two tubes, fine, two tubes like that. Let me draw it, actually that will make more sense. Okay. So, let us move on to, okay, counter current multiplier. Multiplier. Okay. So, what essentially is counter current multiplier? So, let us think of two tubes here. So, let me pick up the right color set. Okay. Something medium. Fine. So, one tube is like this, okay, and there is another tube which is like this. Fine. They are in touch, but they are not continuous with each other. In other words, okay, and out here like this. Okay. Now, think of a situation. So, from here a fluid is moving or water is moving at 0 degree centigrade, and from here water is moving at 100 degree centigrade. Okay. By the time, so automatically the one which is moving at 100 degree centigrade will dissipate its heat to this side, if there is a there is a possibility of a conduction if they are not very well insulated. Okay. So, essentially what will happen, the water which will come out of it is 50 degree here, 50 degree here. In other words, what has happened, the water which has moved at 0 degree has gained heat out here, and the water which has moved at 100 degree has dissipated heat out here and reduced its temperature. Now, think of a reverse situation. So, this is a simple exchange. This is a very simple exchange of fluid. There are two tubes which are moving parallel. Now, think of a reverse situation. So, let us talk about the reverse situation now. Reverse is, okay, one second, pick up the right color. Okay. So, here is one tube which is moving through. Get the tube a little thicker. Fine. Okay. And there is another tube on the side which is in yellow, like this. Okay. Now, we are reversing the case of the direction of flow. What we are doing essentially is, from this side water is moving at 0 degree, and from this side water is moving at, or rather sorry, from the reverse side water is moving at 100 degree. Now, what will happen? This is moved at 100 degree and 100 degree centigrade, and so what essentially happens is that, so while water is entering, it is at 100 degree centigrade, and here water is moving, it is at 0 degree centigrade. From the point it started touching each other. So, up to this it is fine, and up to this it is fine, it is all moving this. But from here on, situation will be something like this. This will be at 80, this will be at 70. Okay. This will be at say 60, this will be at 50, this will be at 30, this will be at 20. At the end of it, this will come out as 10 degree and this will come out as 90 degree. So, this is the phenomena where the fluid is flowing in two reverse direction with different parameters makes change the energy in a different way, and that is what is called a counter current mechanism. And this counter current mechanism is fairly common in biology, and one of the classic case of counter current mechanism which is being followed in the kidney is in the loop of Henle. And now I will show how basically this counter current multiplier, actually it is called counter current multiplier, helps in concentrating sodium and other ions in the reabsorption and absorption processes. Okay. So, what we will do, we will consider these two tubes now, and we will see. So, this is basically a loop of Henle like this, okay, and we will observe how with respect to the interstitial fluid, because here you have the interstitial fluid, you have the kidney, the Bowman capsule, sorry, the loop of Henle, excuse me, loop of Henle which is moving. Okay. So, if there are two arms of the loop of Henle, so something like, let me draw it. So, the situation is like this. Okay. So, here you have the two arms which are moving like this, very close proximity, and you have the interstitial fluid in between. So, this whole part is interstitial fluid, okay, which has a different kind of osmolarity values. Now, what will happen? Now, you have the okay. 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 Fine. Let me redraw this part. Okay. Okay, coming back to the okay. Now, you have here water moving. So, this is the counter current loop of water. So, and values which are being shown are the osmolarity values. Okay. So, out here you have 600, osmolarity 600 becomes 800, okay, and then here you have 1000, 1200, here you have 1000, 800, 600, and what is essentially happening is that, and this is all water molecules here. So, outside it is 600, and from this it is 600. This side also it is 600. Then here you have 800, and you have 1000, and same way from the surrounding 1000, 800. So, this is the counter current loop which is taking place here. They will try to balance each other on either side. So, on both sides it is balancing. So, now if you see how the counter current multiplier works, okay. Okay. Counter current multiplier works like this. As of now, we haven't introduced any. In the counter current mechanism, I never introduce any active transport of anything. So, say for example, it is entering at 400, okay, osmolarity value. So, water. So, here what is happening? it becomes 600, it is getting more concentrated, then it becomes 800, it is getting even more concentrated, then it becomes 1000 osmolarity value. The bottom it becomes 1200, then it again becomes 800, then it becomes 600, then it becomes 400, and it becomes 200, and likewise. So, what essentially happened at this point? So, these are the zones. If you remember, I was telling you that there are energy dependent mechanisms which are governing several things out here. So, now coming to those energy driven mechanism. So, what is happening here is NaCl is being thrown out by an energy driven process. So, when NaCl is being thrown out, okay. So, what essentially happened in that process is, so you have 1000 here, and in between you have 1000. 1000 on this side, because there is nothing is being thrown out on this side, 800, 800, 600, 400. Okay. So, what let me just finish this drawing, so that it started with 400 and then it landed up with 200. It is reducing. So, what is happening? So, and here it is basically milliosmol per kilogram of water, and here you have 1200. Okay. So, in the counter current multiplier, what essentially is happening? Say for example, the water is entering with a different osmolarity, but if it would have been a regular counter current mechanism, which I just showed you before this is a regular case where there are no active transport which is taking place. So, you see 800 here, 800 here, 800 here, 800 here. But think of a situation where out here, where you have these transporters which are sitting here, or this one, this one, this one. So, they are continuously regulating or modulating or manipulating the solute concentration. And so, if you look at it out here, it started with 400 and it ended up with 200. So, there is basically a fall in the osmolarity value, and sodium has been retained. Okay. Similarly, if you look at it in this zone, it is all the same, because there are no transporters. As soon as these transporters comes into play, the things it starts changing. So, this is where the counter current multiplier is one of the mechanism by which kidney ensures that we retain as much fluid as possible. Okay. So, from here what I will do, I will move on to the aldosterone, the role of aldosterone, what it is doing in the kidney in concentrating the urine, and then we will talk about the renin-angiotensin system and we will close in that. Okay. So, coming back to the role of aldosterone. So, say for example, you have a situation when there is a water deficit, you are in the desert or some kind of a situation, water deficit situation, and so plasma osmolarity will be going up because your water is shortage, and your atrial pressure is also go up. Under these situations, what will happen? Your antidiuretic hormone ADH will be secreted, and that will activate the thirst area. Okay. And this ADH which is secreted will tell the kidney. So, here in the kidney, an ADH is secreted up in the pituitary. It will tell kidney for water resorption, this will go up, maximum water reabsorption, and urine goes down. So, antidiuresis. Okay. So, this is how from the brain this is being controlled. And now think of another situation when there is a pick up another water excess. Okay. So, under water excess what will happen, the same thing, atrial pressure AP, that is going to go down, sorry, atrial pressure, so I made a mistake, kindly correct it. So, initially what is happening, your atrial pressure here is going down and atrial pressure here is going up, okay, and your plasma osmolarity is basically going down, and that situation there won't be any further secretion of ADH and there won't be any signal. So, what will happen, water resorption will fall down and your urine secretion will go up. So, this is what is happening with aldosterone route. There is a second thing, what will happen in the salt excess and salt deficit. So, for example, if somebody is fasting, so there is an deficit or somebody is eating too much salty food. So, there is a salt excess. So, if the salt excess is there, so automatically your plasma volume will go up. It will go up because it will retain more and more water. Okay. Once the plasma volume goes up, it inhibits the secretion of renin, which is present on top of the kidney, okay, out here. Okay. So, if the renin is secretion, so sodium resorption goes down. So, you do not need, because you already have excess salt out there. So, you do not really need to activate anything. But think of a situation when there is a salt deficit. This is what happens in fasting. Salt deficit. Under a salt deficit situation, your plasma volume is going to go down and this will lead to the secretion of renin. So, what renin will do essentially, renin will activate something called angiotensin, and this angiotensin will secrete aldosterone. Aldosterone will essentially will just do the reverse thing, sodium reabsorption will be increased and salt excretion will be reduced. Okay. Salt reabsorption will be reduced. And if you look at it in slightly more what exactly happened when there is a renin release? When there is a renin release, renin essentially breaks down angiotensin, and after breaking angiotensin it forms something called angiotensin 1, and angiotensin 1 then transformed into angiotensin 2. It is this angiotensin 2 which has multiple function. This is basically appetite for salt is increased, your thirst is increased, your glomerular filtration rate is being reduced. Okay. Your aldosterone secretion further lead to salt and water retention and ensuring that there is no further loss in this whole process. So, what we essentially see here is that while from the Bowman capsule the fluid moves to the nephron, that fluid has to be processed very, very tightly. You cannot afford to lose too much of sodium, too much of water. You have to retain as much as water as possible, as well as the electrolyte, all the other electrolytes, and you have to ensure that you lose urea and other unwanted materials. So, there is this counter current mechanism which is coming into play. There is this osmotic pressure, there is this hydrostatic pressure, and then we talked about these are being controlled by the higher centers of the brain. So, that brings us to an end of this part of kidney and the regulation of fluid. Thanks a lot.